Good afternoon, everybody. Welcome to this week's Institute for Quantitative Biomedicine seminar. I'm Stephen Burley, the founding director of the Institute, and more recently, the interim director of the Rutgers AI Data Science RAD Collaboratory. And you'll be hearing more about that in subsequent weeks. We have our first data science AI presentation in this series a week from today. Dr. Mingjia Zhu from NJIT. Today we're hosting a friend and a colleague locally. And next slide, please. So before I introduce Zoltan, just let me remind you that we've got 4 additional seminars before the end of this semester's series, culminating on the 11th of December with Romeo Morrow from UCSD. Next slide, please. Another pitch for nominees, applicants for the IQB Inclusive Leadership Award. Please, if you feel you're qualified or if you know of someone who is qualified, please work with Michelle to make a nomination or an application before the end of the month when the opportunity closes. Next slide, please. So we have a series of training events coming up shortly. On the 4th of November, Shuchi Dutta, our Molstar expert, will be presenting a virtual office hour. This is an opportunity for you to learn more about the intricacies of Molstar, which is an extremely powerful system for display of 3D biostructures. Next, please. Later that same day, we've got a webinar that's going to be presented jointly by Sebastian Bittrich from our UCSD team and Dennis Peel from the RCSB PDB here at Rutgers, focused on binary SIF and the advantages that particular compression of PDB data enables. Next slide, please. And then 3 days after that, our Ezra Pasak will be talking about supporting extended PDB IDs. This office hour is largely for the consumption of software and software providers who access PDB data, who need to be in sync with the changes that we're going to make for PDB IDs that'll make the contents of the archive more accessible broadly. Next slide, please. We continue to have a number of openings here, both here at Rutgers and UCSD, listed on this slide. The most important of which, from my point of view, is the search for my successor here at Rutgers. That job was advertised in September in Science. If you know of anybody you think would be a good candidate, please don't hesitate to make suggestions to me, and I'll write to them and ask them to apply. We've already received some encouraging initial responses to this year's advertisement. So please watch this space. Various candidates will probably be coming through in the coming months for seminars and visits, etc. Next slide, please. Just a shout out to the entire IQB team that helps make all of this possible. Please be sure to acknowledge them when you see them and be grateful. We couldn't do what we do. We couldn't be successful without all these people supporting. So next slide, please. So let me close by introducing today's speaker, Zoltan Szekely, who is a member of the Molecular Design and Synthesis Lab here at Rutgers. Zoltan is a native of the country of Hungary, where he received training in both bioorganic chemistry and then subsequently medicinal chemistry, getting his PhD at the Albert St. Georgi Medical University in Szeged, Hungary. Zoltan has many interests, and today's talk is going to span both small molecule and large molecule drug hunting, with the goal of designing novel molecular probes for complex biological targets. So thank you again for agreeing to speak, and thank you all for attending.
Thank you so much for attending, and thank you again, Stephen, to introducing myself. I have to tell you, Stephen keep inviting me for various seminars, CING, proteomics, many times past couple of years. So he should have memorized my resume by now. So let's get started. Today, I would like to give an update on a. Oops, is that me? That's fine. No, it's fine. An update on some antibody-drug conjugate research and some perspective on ongoing small molecule and peptide work at Rutgers with various collaborators. Just a little, okay, just a housekeeping item. I need to disclose that I'm a CTO of Szekely Pharmaceuticals. That's an antibody-drug conjugate company. Rutgers spin-off company founded by the late Joseph Bertino, Stragnoling, and myself about 6 years ago. I will talk about the ADC, the subject of this company, in a minute. Let me try to move on. So the outline of the presentation today, I would like to talk about an M69 MMAE ADC candidate, then peptide-based drug discovery for Ricin and Shiga toxins. And I would like to show a couple of slides on new modalities for cancer therapeutics, and finally, some slides on new technologies, instrumentations being installed at our facility and ready to use for various drug hunting projects. I think I would skip that since Stephen kindly introduced me. I'm at Rutgers since 2010. What are antibody-drug conjugates? It's a very important field of new cancer therapeutics. Okay, here we are. If you have a good cancer antigen, a good cancer target on a cell surface, and good means highly expressed and specific for certain cancers, obviously you could develop an antibody, typically a mouse antibody, then humanize it. With proper technologies, it's possible to link highly cytotoxic compounds which are totally stable in circulation. The linkage is stable in circulation. Then the antibody-drug conjugate goes through endosomal lysosomal processing, releasing the drug, which ultimately leads to cell death. That's the concept in a nutshell. So we have three components: a good humanized, deimmunized antibody, highly cytotoxic, below nanomolar, typically below nanomolar range cytotoxic agent, and a linker which should be stable, but then releasing the drug intracellularly. Recent, I would say 10-15 years, it's an ongoing trend. More and more ADCs got approved by the FDA, 1 or 2 per year. At Centris was the breakthrough antibody-drug conjugate from Seattle Genetics, because finally they could optimize the linker. Their so-called cleavable valent citrulline linker, which is using cathepsin B in the cell, was the first notable ADC back then. And even today, that valent citrulline cleavable linker technology is used in many other ADCs being approved. This is a brief market analysis summary. By 2028, significant $24 billion annual market predicted for the ADC, the anti-cancer ADCs. As you can see, still blood cancers, breast cancer dominates the field, but more cancer types getting into the ADC field. The main subject of today's talk is R-ADC. It's a first-in-class antigen targeting ADC, activated matriptase targeting ADC. Matriptase is a serine protease, which is very important to cancer proliferation, and about over 10 years ago at Georgetown and Rutgers, they could identify it as a cancer marker. Soon, mouse antibody was developed, M69, which we used for ADC development. Now, what are the issues with ADCs in clinical development before we go into details and specifics about our ADC? One of the main problems is that the ADC field is focused on a few targets. We have a shortage of targets. HER2 for breast cancer, for example, a typical target, and TROP2 for also breast cancer and other cancer types. We need to add new successful targets to expand the ADC utilization in cancer research. That's why we are very excited about our new activated matriptase targeting ADC. This is just a small schematics. Matriptase expressed in a latent form, and then by a proteolytic cleavage getting activated, and this activated matriptase is the molecular target, the real cancer-specific molecular target that can be targeted by M69 antibody, either as a single molecule or complex with this stabilizing protein, or HAI1. M69 recognized in both formations, but only activated matriptase can be recognized by M69. That gives us a good cancer-specific targeting opportunity. When we started over 6 years ago, we analyzed the potential chemistries and got to the conclusion that we need a click chemistry-based approach. Why? A typical IgG has about 1,300 amino acids and contains about 80 lysines. That's 6% of the amino acids. We either utilize, that's the classic way in ADCs, the cysteines, the disulfide, which is first partially reduced, put some drug conjugation on the sides, then reoxidized. The alternative approach is using the lysines. We have so many that even if you use a couple of lysines in the CDR domains, it's not that important. We don't lose much of an antibody binding to the antigen. So we decided to utilize the lysines. And back then, we developed a two-step chemistry. So instead of manipulating the disulfide bridges, I was very worried to structure damage the antibody. We decided to use, we decided to use some of the lysines. We knew from the literature that based on the drug, choice of the drug, which is monomethyl auristatin E, a tubulin binding agent, we need only 3 or 4 per antibody. The drug antibody ratio can be about 3.5 for ideal action, optimal mechanism. So we utilize lysines and develop the two-step chemistries. First, we have a short pegylated click chemistry reagent, a DBCO, that was developed by Caroline Bertozzi many, many years ago. And then the second stage is using the toxin with the cleavable linker developed by Seattle Genetics, the valent citrulline linker developed by Peter Center at Seattle Genetics. Again, pegylated and acid-terminated structure can be added as a second step to the activated lysines. Long story short, when you do it right, when you optimize it, you end up with 3 to 4 derivatized lysine per IgG, which is ideal. You cannot use too many because it will crash out, aggregation could happen, and you need at least that many. Even with this highly cytotoxic payload, the monomethyl auristatin E, you need 3, 4 per antibody to have a significant anti-cancer effect. And it's just a quick QC. Our antibody is 147 kilodalton. The average gain after doing all the chemistries is about 7 kilodalton. This unit is 2,000 Dalton, 1,997 to be correct. So the 7 kilodalton average gain of molecular weight gives you 3.5 average payload loading per antibody. Perfect. That's what we aimed. Couple of notes on the pegylation. These anti-cancer agents, not just the MMAE, but many others, are very hydrophobic compounds. So it's always a problem when you link them to protein surfaces. They could cause aggregation and conformational changes. So that's why I utilized polyethylene glycol here and a short polyethylene glycol between A and the protein surface. That eliminated aggregation problems. And the whole thing is now recognizable by the cleavage enzyme by cathepsin B. So it has multiple benefits, biochemical benefits. Later on, Simons from Seattle Genetic published a paper that pegylation between the payload and the antibody-drug conjugate lowers the off-target effects. Pegylation somehow resolved the problem of the drug interacting with the drug interacting with the antibody surface and creating certain molecular features which can be uptaken by hepatocytes, for example. So these days, pegylation almost mandatory for modern new ADC design. We have a Rutgers patent, by the way, about this whole technology, which was just granted last year. I don't want to get into too much details, but you can see that triple-negative breast cancer highly expressed activated matriptase. This cell line has no activated matriptase expression, significant difference in cytotoxicity utilizing our ADC. Same can be seen non-small cell lung cancer, activated matriptase expressing and non-expressing, and a lot of cancers, prostate, mantle cell lymphoma, gastric cancer. That's a big unmet medical need right now. Very significant cytotoxicity observed, ovarian, pancreatic cancer, multiple cell lines proven that activated matriptase is a good cancer target. And of course, we went for animal studies, xenografts. These are triple-negative breast cancer, significant tumor suppression observed. Interesting that we tried the antibody itself, this green line. It cannot be used as a therapeutic antibody like Herceptin for HER2. The HER2 targeting is so complicated mechanistically that even on a non-loaded naked antibody could have enough effect to create anti-tumor effect in vivo. In our case, you need some significant payload, like we did monomethyl auristatin E to have anti-cancer effect in vivo. The animals were healthy, no change in body weight observed, and no significant toxicity observed. Not so optimized, but some preliminary data on lung cancer and prostate cancer. Of course, it was just one dosage, no optimized dose regimen, still anti-cancer effect observed. We did tumor microarrays and found significant expression of our target on many cancer cells, and we used 30 normal tissues from 3 donors, and negligible staining was observed on non-cancerous cells and tissues. Right now, we try to explore new payloads like pyrrolobenzodiazepines or even topoisomerase 1 inhibitors, or redesign the antibody. Bispecific antibodies are gaining more and more importance in the ADC field. These are kind of open-ended developmental plans. Our business strategy is to push the M69 MMA ADC to clinical trial as soon as possible, of course. The next subject are peptides, peptidogrammatic, small molecule drug conjugates, and other related subjects, how we try to extend the existing chemical capabilities and molecular modalities at Rutgers. Just a reminder that peptides are not just molecular probes, not just diagnostics anymore, but they are druggable molecules, and more peptides becoming therapeutic agents. Just think about weight loss medications as an example. The injectable peptide drugs are on the market, and the FDA has getting more and more excited to approve these kind of drugs, not like many years back. I used to call peptides force multipliers in biomedical research because they are relatively easy to make, they are relatively easy to derivatize and make libraries, and even with limited workforce and budget, we could quickly move the project forward and get better and better peptide candidates, a little bit faster than small molecule research. This is just a patent summary and potential market growth in the next, let's say, decade in peptides. Just a recent data from PeptiDream Incorporated from Japan. They had a KRAS targeting peptide, which had a moderate inhibitory activity, not much cell in vitro cell-based activity, but with relatively straightforward changes, keeping the scaffold, but adding some special side chains. They dramatically improved the target affinity and the cellular IC50, and it's an orally available drug in clinical development. So even a relatively large peptide, which would be seen undruggable many years ago, now suddenly become not just a druggable, but an orally available druggable drug candidate. That was published in 2023. So that's one of the recent examples for a great peptide translation from a non-orally available, low cell activity to a very, very promising clinical candidate. So that's the technologies used for that in Japan are all available at Rutgers in our laboratories. So it's just a recent example. Our local example is the Shiga and Ricin toxin project. The PI is Professor Nilgün Tümer at the Plant Science Department at SABS. She's working on these toxins for many, many years, and it's a very important field because Ricin, obviously a biothreat agent, Shiga toxins can be produced by, Shiga toxin can be produced by E. coli, so it's a food safety issue, and they both are unmet medical needs because there are no antidotes or antibodies, FDA-approved drugs in the case of Ricin or Shiga poisoning. I picked that project because it's a good example of peptide-based drug discovery and target drug discovery and drug development. So in a nutshell, hundreds of thousands of metric tons of castor bean produced annually on Earth to produce castor oil, and the leftover material after pressing for the oil is a highly toxic solid which can be utilized as a biothreat agent. It's more than 5% of the seed is the total toxin concentration, so it's relatively easy to isolate and use it. Again, no antidotes or vaccines, and that's why about 20 years ago, the US government started to fund antidote research. The other similar, mechanistically similar toxin is the Shiga toxin. It's produced by E. coli, and in the case of infection, it could lead to hemolytic uremic syndrome, which is quite a serious situation, especially in children. Just a recent news flash, as of October 22nd, an E. coli outbreak linked to McDonald's Quarter Pounders resulted already one death, hospitalization, multiple cases in 10 states of the United States. And these are coming up almost every month, every other month. And it's all going back to E. coli producing Shiga toxin. Mechanistically speaking, the Shiga toxin and the Ricin toxin depurinating the ribosomes, and only one depurination event deactivate the ribosomes, and the toxin could destroy the activity of hundreds, if not thousands, of ribosomes. So that's the mechanistic reason why that's so extremely toxic. These P-star pentamer proteins are responsible to home the toxins to the ribosome surface, and that is the red surface area represented the depurination site. What we try to inhibit is the first recognition space step, and since it's a peptide protein interaction, we try to develop peptide-based inhibitors for the event. As holotoxins, they are not active because either the P-star binding site or the depurination active site are hidden in the case of Ricin, in the case of Shiga toxin. Significant effort was taken place by Rudolph, our collaborator at the New York Structural Center. By 2020, they published a peptide complex with Shiga toxin. This X-ray crystallography included a P11 C-terminus of the star protein. Unfortunately, only the first C-terminus, 6 amino acid, can be determined by X-ray, but that's enough structural information to start peptide-based drug development project. Interestingly enough, although the mechanism is similar, the binding sites are quite different. This is the P11 fragment. The P6 are highlighted in the case of Ricin peptide complex, and this is a shorter version, but again, the P6 highlighted in the case of Shiga peptide complex. As you can see, quite different binding site and different amino acid side chains involved. So the reason I really want to highlight this project, because this is the peptide to peptidomimetic transition is really a sustainable, doable drug development approach at academia. It's not as labor intensive as a small molecule drug development project. Starting points can be bioactive peptides, like in the case of Shiga and Ricin project, or we can utilize phage displays to have a starting point. And then what I want to show you in a second, in the case of the ribosomal P5 peptide, 5 amino acid long peptide, even a simple mutation analysis resulted in immediately higher affinity peptides. So, and when we have these peptides, all the designer and synthetic tools are readily available for a pharmacokinetic ADME optimization here at Rutgers. Unfortunately, I cannot tell you the mutation because we are writing the notice of invention, but I try to. So this is the P5 peptide. This is the wild type, no mutation. You can see 2 key phenylalanine residues as moving while binding to Shiga toxin. Even just mutating one of the phenylalanines resulted significant increase of the binding activity. So now the next step, of course, doing what you can see in the KRAS paper, various cyclization and other modifications happening. It's a good news because these peptides are really easy. Oops. These peptides are really easy to make, and we have collaborators for peptide cellular delivery. Various nanotechnology methods can be used to deliver the peptides to the toxins. Oops, I need to move it. So that's about the Ricin and Shiga project. Next is I try to show a couple of slides about advanced instrumentation. No matter we use small molecule approach, peptide approach, ADC approach, we need to make sure that our synthetic capabilities can support the project. So we can produce enough, let's say, peptides for the Shiga Ricin project. And we're not just synthesizing them, but purifying them and analyzing them rapidly. And after introducing the new chemical entities, we need to make sure that we have the molecular level imaging capabilities readily available to check the fate of the peptides and ADCs in cells and in tissues. And we need to make sure that the in vitro and in vivo models we use are continuous. Let me show you a few slides about that. For peptide synthesis, we are about to purchase a new microwave-assisted solid-phase peptide synthesizer. We have a very old machine. This machine has about 4 minutes per amino acid cycle. So within an hour, you could introduce roughly 12 amino acids, 15 amino acids. That's the future, per se. And even further down the road, we would like to make sure that we could produce small molecule libraries. These are even more costly machines, solution or solid phase small molecule synthesizers that will take an S10 grant from the government. It's about $100,000. These are a couple of $100,000 machines. We have some 30-year-old, my peptide synthesizer is 32-year-old. So that's why we try to catch up with the hardware capabilities. I wanted to take this out, but then I changed my mind. So we have a target. If you have a target, we have some imaging modalities, maybe confocal microscopy, flow cytometry, some ELISA assay to check for the inhibitor activities and other molecular tools. Then we go for cells, organelles, and then finally animal models. This green to red transition shows that at certain transitional stages, we have imaging problems, we have assay problems. We are very good at saying simple, isolated molecular target. A ton of methods and instruments available. But when we go to more and more complex models, our capabilities are limited. That machine would solve a lot of headaches for us, because if we would have a mass spectrometry-based microscope, we could have, let's say that's a brain example, but whatever tissue and organ you target, you could make slides, and this MALDI-based mass spectrometer could ionize micrometer by micrometer the surface of the tissue, create ions, and you could analyze the fate and trap the fate of your molecular target in that tissue. That's just a suggestion that, again, we need to form some kind of user group and go for an S10 grant. Unfortunately, it's about $1 million, but it could be used for cancer, metabolomics, and in this case, neurodegenerative diseases, and so on and so forth. We need to figure out how to get the $1 million for that machine. This is the molecular design and synthesis research facility page. I don't want to get into too much details, but we have plenty of mass spectrometers, purification machines, microwave synthesizers, and all kind of small-scale, large-scale analytical and preparative purification tools for small molecules, and we keep adding up the peptide chemistry to that. What is ongoing now? Recently, we received with Dr. Copeland a brand new high-end plate reader. Also installed a small liquid handler robot for sample preparation, and which is coming up soon, in vivo organ-on-a-chip system for it's a microfluidic chip-based system for organ-on-a-chip type investigations. That was founded by the New Jersey Health Foundation. So in summary, we have a new ADC candidate, a first-in-class serine protease targeting ADC drug candidates, which is a Rutgers patent and currently in development by a spin-off company, Zyconix. We have new modalities available for principal investigators and collaborators, peptides, peptidomimetics. I have a couple of extra slides on DNA minor groove binding agents, small molecule drug conjugates, and we keep introducing new technologies, new instrumentations, and we try to move toward mass spectrometry as an imaging tool applications, and we highly emphasizing the importance of automation in our research, not just in peptide synthesis, but in general, peptide and small molecule research, synthesis, purification, characterization, and assay capabilities should be improved and being improved as we speak. Stephen? Oh, I'm so sorry.
Acknowledgement. Yeah, go ahead.
I'm so sorry.
That's what I was waiting for. Yeah.
I would like to dedicate this lecture, as always, to my late mentor, Dr. Joseph Bertino. He was not just a great scientist, a great stimulating individual, intellectual. We miss him. Our stimulating collaborators, just a few of them, Professor Tümer on Ricin and Shiga toxin, Professor Muradian, Parkinson's disease, Professor Pilch, antibiotic research. They are really stimulating, on a daily basis, stimulating people. I'm really happy to work with them. On the molecular design and synthesis core facility, I would like to thank you for our director, Jacques Roperge, who is keeping moving the facility and keep extending the facility to new chemical modalities. Office for Research, G2 and Michael Zwick also extremely helpful and supportive in our adventure. Zyconix Pharmaceuticals, Vijay Samant, our CEO, try to make sure that this Rutgers intellectual property will go to clinic as soon as possible. And finally, Department of Chemistry and Chemical Biology, without Professor Williams' help, we wouldn't be as effective as we are at this point. We try to make sure that our facility and the department is getting more and more involved in research. And I think that's
Thank you very much. Let's give
you.
Zoltan another hand. You mentioned you have a couple of back pocket slides, or do they follow this?
Yes.
Yes. Why don't, since there is time,
Yes.
I think why don't you show those, and then we'll open it up to Q&A.
So many years ago, when I worked for the NIH, that was a push for some kind of chemical genetics, meaning relatively small molecule. Back then, no ADCs available, no fancy nanotechnology available, just small molecules targeting certain transcriptional factors, try to alter gene expression. It did not work out. That was a company utilizing DNA minor groove binding agents. This is just a natural product. Of course, they had much more sophisticated compounds. GeneSoft, that was a Peter Derwan's spin-off company in California. Unfortunately, when they tried to target relatively longer sequences from the minor groove, they couldn't deliver it to cells. So these days, based on ADC technologies, utilizing other drug delivery systems, that whole field can be should be revisited. Okay, we have the chemical capabilities to make these compounds. We even have the chemical capabilities. It's just an example. It's a summary how many potential DNA minor group binding agent utility can be used to target HIV expression. But of course, various cancer applications would be developed based on this technology. The reason I'm mentioning that these small molecule binding agents are highly sequence selective, and they typically use pyrrole imidazole amino acids. So it's a peptide synthesis-based approach. Automation possible, relatively easy to make them. There are large structures, but repetitive chemical processes can be used to produce them. I did them on solid support many years ago, like a peptide. So that's it's not an active thing, but I would like to emphasize that if somebody is interested in targeting the DNA, we have molecular tools, we have chemical capabilities, we have a patent long back how to make them. And again, based on what we see on ADCs, these drugs or drug-like molecules can be selectively delivered to cancer cells, for example.
So instead of a toxin hitched to the antibody, you would link these molecules to antibodies, target them to cancer cells, and then have them go in and shut down.
One benefit would be the lower off-target effects, because if you lose it by accident, you always use a little payload, no matter how fancy your linker technology. These compounds are non-toxic. They're so big they couldn't even get inside the cell to begin with. But even if there is some active transport happening, they are just not toxic.
And how do they get in? If they do become liberated from the antibody, how do they get metabolized? Is it through the cytochrome P450?
I'm pretty sure that some cytochromes will start oxidizing the aromatic systems. To be honest, I'm not sure. They are, as far as I know from the Peter Derwan's papers back in 1998 and 2000, they never had a stability issue. They had a delivery problem of these.
Yeah. But eventually you've got to excrete them. So the question is, so maybe they just end up in the urine.
Yeah, possible. Okay. So that's one thing, and it's just some structure interpretation. Let me skip these
one of my ex-postdocs did that work as a PhD student a million years ago.
That's really, it was it's a sleeping beauty in biochemistry, but it could be resurrected. So let me. I don't even want to get into structural details, but small molecular drug candidates, and this is just a reminder. So took many years at Bristol Myers Squibb, went to clinical trial, didn't work out very well. Small molecular drug conjugates. Again, we have all kind of new linker technologies now. You have a folic acid. Cancer cells overexpressing folate receptor because they need folic acid for DNA synthesis. An optimized linker linked to vinblastine. And that was a new conceptual molecule. Unfortunately, it stopped about 10 years ago. Now, a company, I think it's Andosite, Indiana-based company, picked up the project, and what they did, they kept the folic acid here, redesigned linker. It's high because folic acid has very low solubility. The vinblastine had low solubility, so they needed a hydrophilic ionic linker. They changed the toxin for this very highly toxic tubulin binding agents, and that was again terminated. But then they kept redesigning, and as you can see, tubulizin was used, and more recently, DNA alkylating, bis alkylating agent a PDB derivative was used. So by tuning and moving to more and more toxic payloads, sooner or later, the folate receptor active transport is able to deliver enough toxicity to the cell selectively. It's a hard game, you know, to balance between total delivery and cancer selective delivery and the toxicity achievable in cancer sense, but it's doable. This is still a funded project, and hopefully we will have some clinical data on these things. And this is just a folate receptor, so.
Yeah. And I just want to say, as a veteran of oncology drug discovery and development, I don't think you should be put off by the fact that most of these fail. 19 out of 20 small molecule drugs that enter clinical trials for cancer never become drugs. 19 out of 20.
Yeah.
So all these small companies with one drug, it's a 95% chance of failure. And that's the grim reality.
But in ADCs, that could be.
Now, with antibodies, it's a totally different story.
Total different story.
Yeah.
Total different story.
So, questions, thoughts?
Thanks. Very nice talk, Zoltan.
So, can we have a little light? Can we? The light, can we turn on the light a little bit, or? But you might need it for the recording. Okay.
So with the, I think the MMAE conjugate, I saw you mentioned a dibenzocyclooctane. Where in click chemistry? So where and how is click chemistry coming into basically the assembly of that? Oh, I apologize, I missed that.
Oh, with your microphone a little bit closer.
Oh, sure. Whoops, blast people. Sorry. So with.
You have 30 people in the room. We've got even more people online.
So with the antibody drug
conjugate, let me show here because it was maybe blocked. Okay, so this is the click chemistry. Okay, all right. Okay, I'm not visible now. Sorry. So this is the DBCO. It's a copper-free click chemistry, and I selected copper-free because you don't want to have even a small amount of copper in the presence of a protein environment if you don't need to. So first, we derivatize the lysine side chains, and then 45 minutes later, we add on the azide. And that's a typical overnight reaction at 4 degrees.
And what do you think is the advantage of having that sort of the cycloaddict?
Yeah, you could, first of all, you could tune the drug antibody ratio because you have a two-step reaction. That tuning is very important. And the second thing is that you could introduce two pegylation, one peg here and one peg here. Back then, I tried to reduce the aggregation effect by loading the drugs. And later on, the Seattle genetics people figured out that, oh, by the way, that works very well to lower the off-target effect, hepatocytes and other cells. So that pegylation is really popular these days. Yes, if you want, you could activate it by an N-hydroxysuccinate or something like that and put it on the lysine side chains. It has better control, more tunability. And if you have excess DBCO, for example, later you could put imaging agents on for research purpose, or radio labeling can be applied later on, either parallel to the toxin or have a totally different product, or a combination of product, which some people try these days, both radio labeling the same antibody and put toxins on.
I mean, just as follow-up is just, so focusing on peptides as therapeutics, where do you sort of fall in the discussion? Because it seems like there's been almost a resurgence of interest in peptides like Cesar de la Fuente at Penn and non-ribosomal and ribosomal encoded peptides.
Yes.
Where do you sort of fall in that discussion as far as their opportunities?
I would think that, for example, the Winfried Vandenborg technologies utilizing the various enzymes for cyclization and things like that can be used to quickly have a second layer of derivatization of your peptide library, And we know that typically a linear peptide, and for many reasons, stability, entropic penalty, sorry, entropic penalty and other reasons, it's not going to be druggable. So you need to have a cyclic, an N-methylated backbone, you know, various things. So basically the post-translational modifications we can learn a lot from nature. I mean, you attended the lecture, Winfried lecture a couple of weeks ago. So that those natural peptide modifications can be mimicked by everyday chemistries. Maybe not all everyday chemistries, but if we have a very complex modification, we could ask him to send us an enzyme at least to do it, you know. So that's I'm very happy that I went to that seminar
and we have a three-part question from an anonymous attendee online. So first, is the PEG linker used in M69 MMAE linear or branch chain?
Okay, it's linear.
Linear.
And more importantly, it's a chemically synthesized PEG. There is a company, Quantitative, introduced chemically synthesized polyethylene glycol. So it's not a polymer.
So it's pure.
Product.
Yeah,
it's absolutely. It's not even mono dispersed. It's discrete. Right.
And then the second question is, what was the rationale to use MMAE versus camptothecin derivatives, given that many companies are exploring
very interesting. And thank you for the question, because I missed that point. So historically speaking, ADCs, the first was Myotarg, you know, then withdrawn, then reintroduced, used calichamycin, almost too toxic and a little bit too big, which is another issue, and hard to link by a stable linker to the antibody. A couple of years later, we're talking about early 2000s, Immunomedics, a New Jersey-based company, tried a new technology, topoisomerase 1 targeting with second-generation camptothecins. They claimed that you could load up to 7, 8 relatively low-potency drugs on an antibody, but you still could get enough toxicity delivered to the cell. These days, it's again back to the more toxic compounds. Right. So if you have a topoisomerase 1 inhibitor, there is Daiichi Sankyo. The company developed a newer topoisomerase 1 inhibitor, DXD. That's about 5 to 10-fold more toxic than the original Immunomedics drug was. It's an opportunity. I would go for the higher toxicity.
Right. So then question number 3.
Less damage on the antibody.
Good. Question number 3, yes, no answer. Does M69 MMAE exhibit bystander effect?
According to the limited animal study and some tumor microarray, I don't think that it's significant.
Right. And then there's a question from Aditya Narvakar. What are the characterization strategies for ADCs versus monoclonal antibodies required by FDA during the approval process?
Oh, that's a great question. Of course, the reproducible conjugation technology, that's an absolute must. You don't need to achieve exactly the same drug antibody ratios, but you need to know, and you don't need to be site specific. You know, these are non-site specific conjugation methods. That's FDA approvable. Most of the, if not all of the FDA approved ADCs are non-site specific. Now there are site specific conjugations coming up. We could use transglutaminase to utilize 2 glutamine side chains, for example. That's a Pfizer technology, as far as I remember. We could utilize the carbohydrates, you know. They could reach up to 2 per antibody. It's a little low, but still approvable. I think the manufacturability, the manufacturing is a key. You need to know, FDA approval. You cannot have free drugs in your product. That's an extremely important thing, because MMA and especially PDB and DM1, we're talking about picomolar cell here.
And no metals either.
And no metals either.
Yeah, right.
Now, metals, that's why I use the Caroline Bertoldi's technology, so there is no copper, no catalyst in this.
Okay. We're at the end of time. I'm sorry, I'm going to have to cut you off. We have to jump to I have to go and give a talk in a couple of minutes, and we have to get Zoltan upstairs. You guys know each other, I know, so please follow up. So let's thank Zoltan once again.
Thank you.